Today we're going to be doing lab 5.3, the Phenol Red lab. This lab helps us detect whether bacteria can ferment specific types of sugars. In this specific experiment, we're going to be testing whether bacteria can utilize ferment glucose, lactose, or sucrose, each in separate tubes. So today I'm going to show you how to inoculate 3 different bacteria and the results and how to interpret the results. So this pH indicator will turn yellow at a pH of below 6.8. 6.8 and below. Okay, so when a bacteria breaks down glucose or lactose or sucrose, the byproduct is actually acid. And there's a lot of different acids that can be formed, like lactic acid, formic acid, acetic acid, and it just depends on what kind of enzymes are used to break down each of these, which pathways are used to break down each of these sugars. So we have glucose here, so it will start out red because of the pH indicator at a normal neutral pH. Then if the bacteria is able to break down, for example, glucose, this media will now turn yellow. Okay, so like orange to yellow. So yellow will indicate that there was fermentation that happened. Another thing that this medium has is a Durham tube. So this is a little tiny tube that's upside down, and what that does is it can catch gas in that upside down tube. This is called a Durham tube. So another thing that happens from glucose breakdown is not just acid, but also gas. So when bacteria is a vigorous fermenter, that means they utilize the sugar very, very quickly and very efficiently. Then the byproduct is both acid and gas. So that gas will be trapped in that upside down Durham tube as a little bubble. That might not always be caught in, the bubble might not always be caught or detected, but if it is, that's another indication that fermentation has occurred, that specific sugar, the bacteria was able to ferment. So now we're going to go through how we actually inoculate these 3 bacteria. So in order to inoculate this Phenol Red, we have 3 tubes. One tube has only glucose as the carbon source. One tube, which is the blue one, is lactose, and one tube is sucrose. So we're just going to take 3 bacteria and we're going to put them into each of these. So first bacteria that I have is some E. coli that's already been growing on this nutrient agar slant. So I'm going to take a little bit of this on my loop. So first I sterilize my loop. Of course, I wait a little bit near the flame. And with aseptic technique, we're going to now take a little bit of this E. coli. And we're going to now put some of it into each of these tubes. So I'm going to take the sucrose tube first. And then I'm just going to touch it a little bit and then pull it out. And then sterilize the loop in between each of these inoculations. So you want to do the one bacteria at a time to avoid any cross-contamination. So we have a little bit of E. coli. Waiting a little bit. Now I'm going to put it into the lactose tube. Again, sterilizing my tube. Wait for the tube, sterilizing my loop and my tube. Wait for the loop to cool down. Grab a little bit of bacteria and place it into the last sugar, which is glucose. Okay. All right, and so you basically do the same thing with the other 2 the other two bacteria. So I'll do the same thing with 3 different lactose, sucrose, glucose tubes with Staph epidermis and also Pseudomonas aeruginosa. So you can see now before the incubation, these tubes are the normal like pH, so neutral pH, about they're like kind of red or sometimes orange. So you always want to keep one not uninoculated tube, so that we can see the difference in that color after incubation about 24 hours. So I'm going to put these along with another set of 2 other sets of 3 for Pseudomonas and Staphylococcus epidermis. And we're going to put this in the incubator for 24 hours, and then we will interpret the results. So after 24 hours, your bacteria of growing, the E. coli should be either very yellow for all 3 of the sugars, and that means that E. coli is a vigorous fermenter of lactose, glucose, and sucrose. And you can see in the Durham tube there, we have a bubble. And that bubble means that not only did it produce acid, that it also produced gas as a byproduct of vigorous fermentation. So this bacteria is able to very quickly and very efficiently ferment all 3 of these sugars. Okay, so we have now Pseudomonas aeruginosa, and again we're comparing to the control. So this control here has not been inoculated, but it does over time, Phenol Red kind of lightens in color. So we always want to keep the 3 sugars as without any uninoculated controls, and we want to compare the colors. So you can obviously see with the E. coli, for all compared to all the controls, it's very much yellow, and that means that acid has been produced because E. coli breaks down and ferments each of these sugars. And because of the acid that's produced from fermentation, that will change the color of the medium because of the Phenol Red pH indicator. So that indicator will change color when the pH changes or decreases. So Pseudomonas aeruginosa is very different. So this one looks a lot like the controls. So nothing, no fermentation happened in any of these sugars for Pseudomonas aeruginosa. So you can see here, glucose is a little bit lighter, but we know that our glucose control was also a little bit lighter. So that's not really a significant showing of any possible fermentation. But what you do notice is Pseudomonas, especially in you can see it really well in the sucrose, it's actually almost getting more red and kind of pink. And so what that means is that Pseudomonas it is growing, because you can actually see the growth of Pseudomonas within this tube. So it is growing, but it's not using any source of glucose, lactose, or sucrose to ferment, but instead it's using another nutrient that's found in this medium. And that nutrient is peptone. Peptone is a source of actually nitrogen, and that nitrogen breakdown is going to cause the pH to actually increase. So Phenol Red, if the pH increases, that actually changes it more of a fuchsia or pinkish color. And so that's why we see a more reddish or pinkish color coming from the Pseudomonas aeruginosa. The next one that we have here is Staph epidermis. So this Staph epidermis is kind of variable results sometimes. So Staph epidermis is showing actually yellow and orange for the lactose. You can see the middle one, the lactose, is actually orange. So if we compare that to the lactose here, so we can see that for sure Staph epidermis has used glucose, And sucrose, okay, so they use glucose and sucrose, and that's why you see this yellow. So what you'll notice though is there's no bubble in the durum tube like there is in E. coli. And that's because staph epidermis does ferment these sugars. However, it doesn't do it as quickly or as efficiently as E. coli, right? So E. coli again has this nice bubble there. These are yellow, but there's no bubble in the durum. So that means it's not as quick. It's not as it's not happening as fast. And so this one is probably fermenting also. If we compare this lactose to the lactose control, they look about the same, but it's possible that this one is also being utilized, but we won't know for sure for that one. We expect that staph epidermis can slowly ferment these three sugars, not as quickly, but it still can ferment. So we would expect that maybe these will be orange or on their way to yellow. It just would take probably longer than 24 hours. If I were to leave this one in the incubator a little bit longer, I can kind of see then if it turns yellow, then I know that, okay, this staph epidermis can ferment lactose. However, maybe it just takes a little bit longer.